Good evening. I'm Hari, and along with Zhen, who we are both part of the bioinformatics team at Latch. And today we're going to be presenting our work on SE Bench, wherein we investigated frontier models' performance on a variety of single-cell data analysis tasks. This was a joint work with Kenny and Hannah, who are right there rooting for us. So without ado, let me get started. All right. I'd like to start with this question, right? So can AI agents analyze real-world single-cell data? Yes. Sure. Well, reliably, let's see. Right, but that's the thought I want to leave you with, right? So basically, we have a ton of data that we've sequenced, and now analytical bottlenecks is kind of slowly beginning to creep up because we're not able to confidently say if the answer is right or not. And to that end, the AI agents have, to a large extent, been able to alleviate this problem, but I don't think we're there yet, and that's what we're trying to investigate with this experiment. So sequencing has definitely improved. The cost of sequencing has certainly gone down, but reliable analysis compresses the data through many fragile choices into a biological claim. And essentially, training these models to be able to confidently say where they can go wrong or like just even start by benchmarking their limitations is something that would definitely help us further how we think about these models. So to that extent, we looked at a variety of single-cell data sets. We had data, single-cell DNA data from Mission Bio's Tapestry kit that also has a protein panel. And we had Chromium data, Illumina's PIP-seq data, which is a single-nuclei RNA-seq data. And data from BD Rhapsody, Powers Biosciences, Evercode, and CS Genetics is single-cell RNA-seq data. All of these have different chemistries, and then we wrote a bunch of evals. I'm going to talk about what evals are in just a bit, but then before then, and then we tested how these tasks performed on a variety of state-of-the-art models from Anthropic, OpenAI, XAI, and Gemini. So at a high level, these models are showing steady improvement in their ability to analyze single-cell genomics data. And then, the general statistical analysis that they're able to do with precision is something that is already impressive. But then our study also shows that there are certain aspects about single-cell data analysis that these models miss, which could be specific to kits or which could be specific to way it handles a single-cell analysis task, which I'll talk about in a little bit. But then at a high level, we also saw that across, as these models evolved, they got better and better in their ability to be able to do single-cell analysis. For example, in terms of accuracy, we saw that there was a nice boost in Gemini's ability to analyze this data. We also saw a moderate improvement in Opus's data analysis skills, and GPT was comparable between 5.4 and 5.5. And in terms of latency, we saw that there was a nice jump in the latency versus accuracy trade-offs for GPT. Pretty much similar for Opus's between Opus 4.6 and 4.7, but then we also saw that the latency did go up by a lot for Gemini. But then this needs to be taken into consideration along with the accuracy that these models were able to bring in. And we also investigated if there are kit-specific artifacts in the bioinformatic capabilities, and we clearly see that the variance can still be broken down across these kits. For example, if you see Tapestry, which is right there at the far right of this graph, the model accuracy is pretty much low compared to the median performance, and this is primarily because this is an entirely different kit. This is a single-cell DNA kit, that has very specific needs in terms of the problems that we are trying to solve with this kit. And this also kind of shows that a lot of these models have been exposed to single-cell RNA, and then there's a lot of work that needs to be done with regards to analyzing DNA data. One thing that still I would like to double click here is that we've been comparing this across different data sets with different complexities, and those are not really comparable across these models. So they could also be that some of these are more complex data, but then despite that, we still see kit-specific artifacts we were able to observe when we analyzed the trajectories from a lot of these models. Right, so one thing that I've been often talking about right here is that, okay, well, we designed evals. We designed, at least based on the previous slide, about 200 evals. But what are these evals, and how do you know an eval is actually good? So when it comes to designing these evals, there are three key design principles that we almost very sincerely stick to, and the first one being durability. So what makes an eval durable, right? Because when it comes to analyzing these data sets, the same inference can be reached through multiple means, and we need to make sure that the eval allows for variation in methodological choices, or that it shouldn't be that it works only with specific parameter choices. So it should have this room for all the right combination of parameters, and still admit these values as opposed to ones that do not make sense. The eval must also be anti-shortcut, or leak-proof. It really should not disclose the answer. Whatever we're trying to test. Essentially, let's say, for example, if you're trying to test an agent's ability to do cell typing, you shouldn't have a column called cell type annotation in your data set, right? That defeats the purpose of basically the model can just cheat. Or sometimes you can also see that you would be too procedural, and you describe the entire method, allowing no room for improvement, or no room for reasoning for the model. So this is what we refer to as anti-shortcut, which is a second key design principle. And the third one is that it's verifiable. So independently, if I give it to a scientist, they should still be able to recover the ground truth and the tolerance values that we've set for these evals. So essentially, the numbers shouldn't be arbitrary, or should not really depend on arbitrary choices for that particular eval. So using these three design principles as governing principles, so we've designed about 200 evals across all these models, across all these kit families, and then we tested these on the state-of-the-art models. So now, I've summarized a lot of these statistics before, but then I'd like to take a deep plunge into a couple of these evals that we thought were very interesting, and we'd like to present to you guys, and then, really show you where the model differed from the actual ground truth itself. So the first one is, I would like to start with Tapestry, so which is a single-cell DNA kit, and I would like to expose the reasoning gap. I'd briefly describe what the eval does, and then from there, let's see what the ground truth looks like. So what we queried the model was, we basically gave it a Tapestry data set, which again is a single-cell DNA and a protein. It's got the protein immune panel. And then we basically asked it to identify the somatic DNMT3A mutation. And then look at the mutant fraction between NK cells and CD4 positive T cells. And all it had to do was compute this and then report this ratio, and then probably answer a biology question based on what this ratio actually means. Let's see what the agent actually did, right? This data set had 5 samples. So essentially, there was one sample where this particular mutation was a germline one, and there was another sample where this one was a somatic mutation. So the test is really if the agent can really look at the data and say, well, this is somatic and this is germline, and then compute this fraction. But then when we actually tested this on the on 16 episodes, or 16 different initializations of the same eval, we found that 11 of them failed because they were not they picked the sample that had the germline variant and not really the somatic mutation. And 4 of them failed because they got the ratio right, but then they were not able to resolve the biology. So essentially, in this case, it actually got the method right because it was able to identify the cell types correctly. It was able to look through the metadata and understand what fields were present in the data set. But where it failed was its inability to really distinguish between germline and somatic mutations with a lot of conviction. So now this is a very clear failure mode in terms of an agent's inability to really process discern between a germline and a somatic mutation. Another example that I have for you is where it performs the computation right, but then it kind of misses the context on why we have the agent perform a specific computation. This is from a PIP-seeker experiment. So this particular study that we were looking at had both single cell and single nuclei RNA-seq data. And for each of these data sets, we wanted to estimate the percentage of cells that are genuinely neuronal. Essentially those are sensory neurons, and we wanted the agent to report this as a true fraction, true neuron fraction between the two kits, right? So it's pretty clear. You'll have to do cell type annotation and then report what fraction of the cells are true neurons. But the lesson is that what the agent used was SNAP25, which is a textbook neuronal marker, but then it also did not account for ambient RNA contamination. What a domain biologist would have probably done is when they saw that there was a high fraction of neuronal cells, they would have gone back and then independently tried another marker gene. So in this case, when we tried nuclear markers with RBFOX3, we were able to see that it was only about 1.6% that were neuronal. And that is kind of very clearly demonstrated by the UMAP that I have right here. So now, when we tried this on 3 different random initializations, we saw that the agent always fell to this trap, and it overestimated the neuron fraction in this data set because it was just restricting to one particular marker and did not really look through the other markers. So now I'd like to pass it off to my colleague who has a few other examples.
Thanks, Hari. Cool. So just to recap, what Hari just mentioned is that for single cell bench, we captured a wide range of assays that have different variability in terms of performance, specifically for single nuclei RNA and single cell DNA analysis. They typically often require, different pre-processing analysis step compared to the traditional single cell RNA-seq. And then for traditional, the widely used single-cell RNA-seq kit, we have Chromium and BD Rhapsodies that are heavily represented in the public, benchmarks as well as open source tools such as Surat, ScanPy tutorials, and widely used in publications. So our hypothesis becomes, for this greater exposure of these analysis in training data, they actually may contribute to stronger agent performance on standard single-cell RNA-seq tasks. So the next 2 examples I'm going to share is essentially expose the agent weakness in terms of these overrepresentation. So the first example comes from the using the 10x Chromium dataset. So for the task prompt, I give it a human 10x Chromium single-cell RNA-seq from small cell lung cancer tumor across multiple lanes from Chromium. And then the question is, ask it to perform pre-processing or appropriate QC step and characterize barcode retention. So this is what the data look like across different lanes. So if you actually look at the data itself, we actually form a bimodal distribution. So one of the lanes actually contain 40K cell-called barcodes, and on average, the other lanes actually have around 2K to 6K range. So this, essentially the 20K cell-called barcodes, they're actually ambient droplets. That's very specific to 10x Chromium kit. And so my hypothesis is whether the agent first is able to look at the data distribution and then make, reasonable judgment in terms of the threshold. So now let's look at where the agent fall. So all 3 agents actually detected a bimodal distribution, but it actually still applied a single global QC threshold. So in this case, it should actually look at the distribution of each lane and then decide, what is the most appropriate threshold, and instead of just do this one cut fits all approach. And one of the run actually utilized the ScanPy tutorial default, which preserve 99% of these ambient droplets. And the vertical line you see here is essentially a ground truth we set, where it should filter out most of the ambient droplets. And then this is another look in terms of the data distribution. So for a scientist who look at the data the first time, to them it's like a big red flag, right? So this is very surprising that agent just used this tutorial approach to handle the data. Okay, so the second kit I choose is the BD Rhapsody. So this is slightly different from the 10x Chromium. Essentially, 10x Chromium, sometimes I think the data I pick is the whole transcriptome. For this case, it's the targeted panels. So it includes around 300 genes. So for this, I want to test if the agent has this like panel awareness to perform analysis. So the task prompt I give is, given this single-cell RNA-seq from baseline triple negative breast cancer monocytes, and identify the top 20 transcriptional shifts between responders versus non-responders after treatment. And so the trap here is that because it's a panel-based data set, not all the genes in the panel are actually expressed in the samples. So in this case, there are a bunch of genes that just have very low noise level expression across all samples. And then what it did, so I want to test to see if the agent is able to pick up the panel-based panel signal, and then see if it's able to pick a good, reasonable statistics to actually rank these top 20 genes. So if it just use the raw log2 fold change to compare, then obviously some of these very noise level genes will be inflated in terms of the fold change. And what the agent did is actually fall exactly into the trap where it didn't look any in terms of the expression across different genes. What they did is just rank all the genes based on the ScanPy log fold change function, and then just pick the top 20, which essentially are just noise and not like very expressed across all the samples. So essentially, it failed to capture the real monocyte signature. Okay, so those two examples actually show that because there are a lot of overrepresentation of these like 10x Chromium or Surat single-cell ScanPy-based tutorials, which influence the choice of the, for example, QC. So now we actually look into the accuracy breakdown by task category. So in this case, for a single-cell bench currently, we have, for example, we break evals into QC, dimensionality reduction, clustering, cell typing, and differential expression. So our hypothesis is that while agent typically actually would fall miserably in the more downstream analysis, such as cell typing or differential expression, because they require more like domain biological knowledge. But from our data, interestingly, you can see that the QC actually show lower accuracy. I think that's actually expected just because the nature of SC bench have included different modality in terms of assay types, right? So they all require their specific quirks in terms of the QC processing, which shows that agent doesn't have these like kit awareness to apply sensible QC for these specific kit. Cool. So with our current design principles of SC bench, so they are designed into these modular categories, as I just showed, so that we actually can be more can essentially test these agents' behavior in a more controlled way. So for example, with the QC, clearly we can just ask you to perform QC and see how many cells are filtered out. And then to cell typing, then we just give it a clean data and then ask it to do cell typing to see if the cell types are sensible. But this also comes with its limitation because ultimately we really want to test the agent's ability to kind of answer the big biological question, right? Given raw data, can you perform end-to-end analysis, pick sensible approaches, algorithms, parameters to derive the actual correct biological insights? So this actually motivates us to work on single-cell bench V2, which is like focusing on the long horizon evaluation tasks, where we give the agent the raw task and ask it like a very high-level biological question to see if it's able to recover true signal. So this is just a little bit like flavor in terms of examples. So what the eval would look like is that you just give the agent a clean, for example, or messy cell by gene matrix, the raw data, and then you ask it, do cluster T cells enhance tumor killing versus singlets? So this is a very high level question, right? So we can actually break it down even more. So then to study the mechanism to understand this question, we can ask something like, so are APC conjugated T cells more exhausted than tumor conjugated T cells? And then break down even more to other different questions. We can actually test the analysis steps. So this type of questions come with its own challenge. So with the breaking down into modular category approach, most of our evals actually use the numeric metric as grader to grade whether the agent did the right thing. So in the QC example, I just ask it to perform QC and see the number of cells that's retained, right? And then see if it makes sense, depends on tissue type or data. But then since now we frame our question in a more high-level, open-ended way, now how do we grade this becomes slightly more challenging. And what we found is that with the numeric grading method, this actually is also very challenging in terms of setting the ground truth. Because first, you gotta find a good differentiator that can actually have a strong signal to show you whether the agent did the right thing or wrong thing. So for example, we're looking for signals like fold change, right? If the agent did the wrong thing, then maybe the fold change is like inflated or vice versa. So with that, how do you set a tolerance window? Now we have agents help. You can actually try to ask it to sweep different parameters, combination of tools. But then if you pick the wrong differentiator, then maybe it's very method variant. So different methods might give you like different answer, which makes your eval not durable. So now with this grading system for the long horizon task, we're more thinking about to use more controlled vocabulary. So instead of grading it purely based on numeric numbers, can it actually answer like final scientific conclusions using these more controlled vocabulary. So this is just still work in progress. So we're still thinking about how to best set up graders for each of the questions we're trying to ask. Still work in progress, and stay tuned for the exciting results. Okay, that's it from us. And thank you for staying till the end, and we're happy to take any questions.
I guess it's like I said that to the protocol and check at the end whether the results published by the agent correlate with the results published in the article.
Yes, exactly. So that's actually what we've been doing. So for all the questions, all the evals we currently built or to test the agent's performance, we trying to anchor it based on publications. And then that actually comes with its own challenge, because a lot of these like publicly available data can also be messy. Some of the paper results are not reproducible.
Yeah, we've all been there where a reviewer too wanted us to add a figure. So like, do you take that? Do you test on that or not? Again, it's a judgment call.
Yeah, so what we've been doing is actually trying to reproduce from our end, perform end-to-end analysis to see where's the gap between our analysis versus agents and authors' analysis, and find the ground truth.
This is great. And for the best case scenarios, what is the reproducibility limits? Do we have a...
Right. Yeah, that's a good question. So in terms of reproducibility, I think that's also kind of tied to the long horizon task, where the downstream ultimate science is pretty similar. It's up to like how strong the signal is, or like the intermediate step. You might get different QC like QC cell counts, right? But ultimately, it doesn't really affect your downstream results.
It again ultimately depends on that particular paper we are looking at, and so we do try to reproduce all the results. And if it doesn't convince any of us, we just that's questionable, and then we move on to the next one.
Yes.
Thanks. This is a great talk. Super awesome work. How do you guys think about assessing the difficulty of the task? Right. So, like, if you're trying to, like, for example, do cell type ID on like PBNCs, for example, it's way easier than doing like subsets of T cells. Right. So, how do you guys kind of parse that?
That's a brilliant question. Like, for example, brain is overrepresented a lot in the literature. So I think agents tend to do get that right. But then on the other hand, if you're looking at pancreatic tissues or liver tissues, you have like nice zonation patterns and things like that, and you kind of use that as guidelines too. So I think it really depends on what tissue you're looking at and what task you're trying to do at the end of the day. For example, I would say the DNA data that we analyzed, although it's a very simple assay, it's a hard problem for an agent because it does not have a lot of things that it could have access to, and it should rather think logically before it tends to answer a question. So I think a lot of these times, it really depends on how well a particular task is overrepresented and how, if a model can simply do it, without a lot of help.
And on top of that, like for cell typing, right, itself is, like there's so many ways. Maybe they're marker-based, there's like reference, label transfer, can you pick the right reference? I think we just open a new avenue. In terms of self-typing, there's different level of difficulty, as you said. So this is definitely something that, like with long horizon tasks, is something we want to explore, is essentially, can agent utilize these, reference, pick the right reference for single cell to do these sort of more sophisticated cell typing tasks. Yeah.
I'm thinking that, isn't biology the only way you will assess it, in the sense that the paper, I'm saying something that I'm not sure it's true, but maybe using agents, you can actually, on an older paper, you can find results that they're more correct biology-wise, right? So is there another type of metrics you can have?
Oh, absolutely. So I think papers could be wrong, and they could be two papers that disagree on a single idea. So a lot of these times, I think there's definitely lots of gray areas when it comes to writing evals that are like super grounded in biology, and which is why we have a bunch of domain experts who we consult with when it comes to producing these evals. So yeah, I think that's a very fair point, and which is where I think you'll use your scientific integrity when it comes to answering things.
Create database or resources that they've been created for years could also be a good.
Definitely, yes, yeah. Yeah.
One more then. Will it be released to the community? Or is, I don't know, because it's private, right? Will you release a version or something, or guidelines?
The benchmark?
Yeah.
Subset of it's available. We don't want the frontier labs to train on it.
Okay.
If you're using it for academic purposes, we can give it to you, and we'll just make you like confirm you're not going to give it to them. That's kind of how that.
Non-profit?
Yeah, if you want it, we can give it to you. Cool. Thank you, guys.